Okay, class, Dr. E again. We're going to continue our lessons on evolution. And in this lesson, we're going to focus on the question of how do we study things in the distant past? How do we determine the ages and the sequences of the existence of different species? How do we know the age of the first rocks? We said that the oldest rock that has been dated is 4.375 billion years ago. How do we know that? How do we know when dinosaurs lived and when they became extinct? How do we know about other extinct species, trilobites or the woolly mammoth? How do we know that different species are closely related to each other in various ways and one may have evolved from the other? So what are the techniques? What are the tools that scientists use? I think it's healthy to ask these questions, to wonder how we determine these things. I'm from Missouri myself, and so I like to have evidence for things. Now you might say, what does that mean, I'm from Missouri? Well, it's because I've seen my birth certificate and it says I'm from Missouri. I've seen the evidence. The types of studies: fossil records, biochemical evidence, radioisotope dating, vestigial and adapted organs. We'll talk about some of these examples. We'll talk about a couple of these in this video lesson, and then we'll continue on into the next. But let's talk about the fossil record. Those organisms that have hard shells or exoskeletons or bones or teeth, these materials can remain intact under favorable conditions over a long period of time, and they become what we call fossils. In addition, fossils may be trapped in tree sap or ice or sediment, but those soft-bodied life forms, like bacteria and worms and jellyfish, will rarely leave a fossil record because they just simply decompose. So most of the things that we have fossil evidence for are those things with hard shells or skeletons or exoskeletons. And it's been estimated that only about 1 out of every 10,000 species has been identified via the fossil record. That means that we do not even have fossil evidence for most species that have existed. An analysis of the fossil record shows that most species that have lived on planet Earth are now extinct, and that the average lifetime of the species is about a few million years. Now, in studying the fossil record, we take advantage of the layering of rocks and of the fossils that are buried in these layers of rock. Or layers are also called strata. And so this technique is called stratigraphy. And the right just shows an example where using logic that the more deeply buried a fossil is, it must have been the older of the fossils. So you have two examples of fossils, one buried more deeply than the other. Now this can show the relative age of species, that is, which died first, but it doesn't give you a determination of the number of years. For that, you have to supplement with other methods such as radioisotope dating. Now this is analogous to my office. The oldest report or magazine is at the bottom of the pile, and I call this a destigraphy. Now, we said that we have to supplement fossil records and stratigraphy with radioisotope dating to get estimates of actual age. Let me give you an example of what we're talking about with isotope dating. Certain isotopes of certain elements. For example, the element carbon has an isotope called carbon-14. Carbon-14 and other radioactive isotopes can undergo a spontaneous decay reaction. Carbon-14 undergoes a decay reaction to nitrogen-14 and emits something called a beta ray or beta particle. And those isotopes that undergo spontaneous radioactive decays have a characteristic half-life for this process. For example, with carbon-14, if we start with a sample of carbon-14, after 5,730 years, half of it will have decayed to nitrogen. So if we take a sample that we suspect contained carbon and we measure the amount of carbon-14, if we have only half of it remaining, we can determine that it must have been something like 5,730 years old. And this is useful for dating human artifacts. Other radioisotopes that are useful for dating things that are much older than that are the isotope uranium-235, which has a half-life of 710 million years, uranium-238, which has a half-life of 4.47 billion years, and rubidium-87, which has the longest of these half-lives. Now, the bottom right hand shows an illustration of how this works. Let's say we have a mineral deposit, and that mineral deposit contains some atoms of a radioactive isotope, and it's a little red star in this drawing. And so at time 0, we have a certain number of these red stars, of these radioactive isotopes. If we were to then sample that mineral deposit, one half-life, and that might be, depending on the isotope, might be like 710 million years after it came into existence, only half of those radioactive isotope atoms would remain. The other half would have been converted to the daughter atom, or whatever the radioisotope is converted to. In this case, it's turning into a green star. And if you two half-lives, only 1/4 would remain, and the remaining would have been converted to the daughter product, the green in this case, and 3 half-lives, etc. So by either measuring the decay of the radioactive isotope, the red star, or the formation of the daughter product atom, the green star in this case, we can measure the half-life of that sample. And the bottom shows a link to this fairly recent study in which a rock from Australia was dated to be 4.375 billion years old. Now, there are other dating and paleontological methods. We will talk about a couple of these in the next set of lessons. One method is what is called amino acid racemization. Now, that's a complicated technique, but you may recall that we described amino acids as existing as either L, left-handed molecules, or D, right-handed molecules, and that the amino acids that make up proteins are almost entirely the L variety of amino acids. Well, these amino acids can very, very slowly be interconverted from L to D. It's called racemization, and that conversion of L to D occurs with a half-life of about 10,000 years. So if you take a very old protein sample, you can determine its age by looking at the ratio of L to D amino acids. Another method involves looking at the rate of mutation of DNA. For example, if you were to study the genes, the DNA for some proteins that are found in most species, such as cytochrome C or hemoglobin, by measuring the rate of mutations of certain amino acids or the genetic code for those amino acids, you can determine, or you can estimate the age at which this mutation came into existence, or in other words, get an estimate of the age of the organism. Also, mitochondrial DNA. Remember, in eukaryotic cells, we have this organelle called mitochondria. Mitochondria have their own DNA, and that DNA is actually inherited from the mother of the offspring. And the mitochondrial DNA mutates more slowly than the rest of the DNA in a cell, and so it can also be used for determining age. Ribosomes, which are made of RNA and protein, are some of the oldest subcellular particles, and they have been very highly conserved over time, but they do undergo some slight mutations with time, and so they can be used to determine the age of very old life forms. Dendrochronology is another way of determining age, which you're probably familiar with if you look at the background of this slide. It's counting the rings on a tree, for example, so that is a method. The magnetic orientation of the Earth's magnetic pole, which we talked a little bit about in earlier chapters on geosciences, can be used to determine the age of the Earth's crust. Another method is the isotope ratio of oxygen in ice, in the water and ice. It turns out that water, H2O, it turns out that the oxygen atom of water, H2O, can exist in either of two isotopes, oxygen-16, which is by far the most prevalent isotope, and oxygen-18. And by looking at the ratio of the oxygen-18 isotope to the oxygen-16 isotope, scientists can get an estimate of temperature in that ice. And that's because the two isotopes, or the two forms of water, H2O with oxygen 16 and H2O with oxygen 18, have slightly different properties, and the formation of ice from these would be a reflection of the temperature, of the ambient temperature at that time. So these are some of the methods that are used in dating both biological materials and geological materials to try to determine their age. We will again pause, and in the next slide we'll continue and we'll talk about some more molecular type of methods, like looking at DNA sequences and how we can use that to determine the relationship of species and when there may have been evolutionary splits between different species. See you in a little bit.